James, and I'm here to talk about my study system. So it's widely acknowledged that animal coloration is incredibly ecologically important. Your color patching can help you attract a mate, avoid a predator, can help you maintain your body's homeostasis. When we combine this with the fact that it's both quantifiable non-invasively and is conspicuously affected by natural selection, it makes it an ideal system to study the evolutionary mechanisms which generate and maintain biological diversity. And as they tend to be small, soft-bodied, and often toxic, color is particularly important for amphibians. Far from being dull and drab, as many people like to think, amphibians can actually display incredibly complex color patterns. And across the group show examples of almost all known color patterns. Some lineages, in particular, are renowned for their color diversity, including the Palearctic salamander lineage Salamandra, also known as the fire and alpine salamanders. Now within this group, their color patterns and this remarkable diversity that they show is particularly adaptive for predator avoidance, particularly in terms of warning coloration, as they secrete a pretty potent toxin, but also potentially for thermoregulation at high altitudes. And really interestingly, within different populations, it appears as though the same color phenotypes may have evolved independently repeated times. So the first thing that I'm trying to do is identify whether or not parallel or convergent color phenotype evolution has actually occurred within Salamandra. I'm doing this predominantly through phylogenetic analyses. Unfortunately, these are taking a while, so it's still a bit of a work in progress, but we do already have some evidence for the independent evolution of certain color morphs. So this is an analysis we ran where we looked at the shallow phylogenetic relationships between the 6 currently recognized species within this genus. And what you can see from the color cartoons on the side is that we have this melanic morph evolving apparently independently at least 3 times. We actually see trends towards a loss of yellow in lots of other lineages as well. So at the moment, we have analyses running with 233 samples from across the phylogenetic and geographic breadth of Salamandra, so that we can really trace color character evolution in fine detail. But for the moment, we're just going to focus in on a particularly color diverse group in this lineage here, Salamandra, to help us achieve 2 main aims. Because we really want to try and objectively quantify color phenotypes within this lineage, particularly using several different methods. And we also want to try and identify genomic loci which are significantly correlated to these different color phenotypes, using both genomic and transcriptomic approaches. And the reason that we're doing this is because to really understand color evolution in the group, we need to understand the mechanisms and drivers of color variation. This is going to help us establish whether or not we're looking at parallelism or divergence when we get that analysis finished. And the reason that our focal population is so good for helping us achieve this is the sheer chromatic diversity that it displays. So across the genus, Salamandra is renowned for being very color diverse, but each population tends to be a little bit samey within that. But that's not the case with our focal population here in northern Spain, where we find all of these different color morphs existing in sympatry. Now, traditionally, these have been broken down into 3 primary color morphs. So we have a brown morph, an ancestral yellow-black striped morph, and a completely yellow morph. However, as you can see, it is a climate of variation. So we're wanting to try and quantify these color phenotypes a bit more objectively. And the first way that we're doing that is to look at the cellular variation that's actually present within these different colors of skin. And we've done that using transmission electron microscopy. And what we found is really interesting. Because within the yellow skin, we're finding that in the top layer, we have these yellow pigment-containing cells called xanthophores. And in the dermal layer, we're finding these light-reflecting platelets called iridophores. But between brown and black skin, we're actually finding incredibly similar cell structures, because in both layers of their skin, they both just present these dark pigment-containing melanophores. But obviously brown and black are different colors, and they are presenting different morphs. So the next step is to try and see exactly what wavelengths of light these different cell complements are actually reflecting. So we use reflectance spectrophotometry to measure the exact wavelengths of light that are being reflected at different point locations on the salamander's body. And what you can see here are just some example spectra for yellow, brown, and black skin. And again, what you can see is that brown and black skin are incredibly similar. They're just differing in the exact amount of light that they're reflecting. But what's also really interesting is that a lot of this variation is being captured within the visible light spectra. There's not terribly much happening outside it. And that's really good for us because it means that we can then use digital photography to look at whole color patterns and not just these point locations. So within our focal group, we took standardized dorsal images from 80 individuals, and then we analyzed their color patterns using this really cool new package called Patternize, which allows you to analyze patterns in multiple ways, including producing principal components analyses. And what you can see from our analysis is that we're getting a really clear separation on this main and by far most important axis of variation between our brown, striped, and yellow morphs. There is, as you'll note, some overlap, but that's pretty good because we do have a kleiner variation. And what we decided is that these PC1 scores are actually giving us a pretty good indication of the color phenotype, better than we could do by just trying to rank them. So what we've done is extract these PC1 scores, and we're using them as the phenotype input for our genomic analyses. And these genomic analyses are really important because when we're looking at the genetic basis of amphibian coloration, we actually don't have that much data. It's not something that's being studied particularly well. So we're trying to infer the genetic basis of color in Salamandra. And the way that we're doing it predominantly is through a double digest rad sequencing approach. So we extracted DNA from the same 8 individuals that we photographed. We created a DD RadSeq library. We sequenced it, and we ended up with a final data set of 2,149 SNP loci. And when we look at just the structuring within this data set, we're not really seeing any structuring based on color, which is pretty good as far as we're concerned. Because now we want to go in and try and find which of these loci are significantly correlated to our different color morphs. And we're doing that through 2 phenotype-genotype association analyses. First, random forest analysis, which is a machine learning technique, which essentially looks for the cohort of covariating loci, which explain the most amount of variation in your data set. We're also using LFMM, which is a snip-by-snip approach, basically based on linear regression. And again, we're using those PC1 scores as our phenotype input for both of these. And we found some pretty interesting results. With the random forest analysis, we identified 9 covariating loci, which explain the most amount of variation within our data set, just over 56%. And using LFMM, we identified 8 SNP loci, which were significantly correlated with our color phenotypes. Luckily, we got some overlap in these data sets. And really interestingly, we got this one, which is really important for us because it's the one that we can most solidly associate to a known color-related gene, TYR, which is important in converting tyranase into melanin, which is the pigment we find in these dark pigment-containing cells in melanophores. The other SNP loci were a little bit less clear. Through various paths, we're able to identify them to multiple potential candidates, most of which are either known or suspected to be involved in animal coloration. But we have a bit of work left to do to find out exactly what these different loci actually are. But we're actually pretty happy with these results because when you're using a RadSeq approach with salamanders, you have a pretty big problem. And that big problem is the size of their genomes. So Salamandra have a genome size about 30 gigabase pairs in size. For a salamander, this isn't that big, but it is about 10 times the size of a human's. Which means that when we're using RadSeq, we can only sequence a small amount of the genome. There are very few existing resources, so we aren't sure what we're going to get when we went in, and we're really excited that we're actually able to pick up some signal of the coloration in this data set. But because we knew this going in, we also decided to pair it with a transcriptomic approach to use as a complementary method of identifying color loci. Because this is allowing us to look at differences in gene expression in different colors of skin, and we used a paired approach so that we could look at differences both within and between individuals, particularly because we had 2 colors of skin on these striped individuals. So we used RNA-seq, we sequenced the gene expression in our tissues, and then we did pairwise differential gene expression analyses in DESeq2, again in R. And from these pairwise comparisons, we identified 220 differentially expressed transcripts, which we were able to associate to 169 specific genes, 58 of which are either known or suspected to have an important role in animal coloration. Which again is really exciting for us because as far as amphibians go, we're not really sure what's happening at the genetic level when it comes to their coloration. And these heat maps pretty simply just show the differentially expressed transcripts in the 2 different, in the pairwise comparisons. And within these 58 genes that we know are related to color, or we suspect, we're finding some pretty common ones that people might be familiar with. You should all be familiar with this one because we also find it in the genomic analysis, which makes me very happy. And these ones are all genes which their either up or down regulation is particularly associated with this yellow coloration. So the last thing I'm going to really discuss is we've started to do some work testing the ecological drivers of these different color phenotypes. So within Salamandra, their different color morphs are predominantly assumed to be a warning signal, so an aposematic signal to predators because they produce this toxin. But it's not been very heavily tested. And we have a pretty good system here where we have brown, yellow, and striped morphs, all very different, to test whether or not there's a relationship between the metabolomic profiles of their different toxic secretions and their color. So we used mass spectrophotometry to look at the compounds present within the toxic secretions. We identified 18 compounds, 4 of which are known to be salamander alkaloids, which is reassuring. But the really interesting thing is that even though we found a lot of variation within this data, we didn't actually find any that were significantly correlated to the different color phenotypes. Apart from in one case, Salamandra, which if you strip out the striped individual, we do have a significant difference between our brown and yellow morphs. So we still have some more work to do examining this further, but it's just a really interesting first look at a test of this supposed aposematic function of this coloration, because we're not finding much difference, at least yet, in the actual toxicity of the animals. So I might have gone a bit fast, just to summarize what it is we've been doing so far in this project. We've been assessing the parallel evolution of color phenotype. We've been able to identify and objectively quantify these phenotypes within a pretty diverse focal group. And through this focal group, we've also identified numerous color genes which are important in animal coloration. And this is really good because it's contributing to our understanding of color genetics in amphibians. It's allowing us to look at the genetic parallelism or convergence in Salamandra, not just the phenotypic parallelism. And it's also allowing us to look in more detail at the supposed ecological drivers of their different color morphs. So there's a lot of work here, and I couldn't possibly have done it all on my own. So I'd just like to acknowledge all of my collaborators, particularly all of the people in Glasgow. So my 2 supervisors, Catherine Elmer and Barbara Mabel, who've been very supportive in this. My collaborators elsewhere, mainly spread throughout Europe, predominantly in the University of Braunschweig. I've had multiple students and volunteers who've helped me work out some of the very difficult and frustrating parts of trying to get this project up and running. And in the microscopy help, I've had some really important collaborators there as well. And I'd also just like to acknowledge all of the funders. I didn't realize how many people had supported this work until I put this presentation together. So thank you for listening. I'm happy to take questions.
Are there any non-toxic mimics around there that might be reducing the usefulness of looking toxic?
No, there are other salamander species there. They look very different, no, there's nothing around there that is mimicking, and I don't think throughout the entire range there's anything that is following mimicry. So yeah, that shouldn't be playing a role.
Any way you can integrate the phylogeny into your gene expression analysis? You're doing pairwise comparisons.
Yeah, potentially. So one of the reasons that we haven't looked at that in great detail yet is that we're still getting the phylogeny up and running, but it is something that we've considered doing. But at the moment, we've just been doing pairwise comparisons. At the moment, because it's kind of exploratory, our sample size is quite small as well, but working with our collaborators, it is something that we've considered. We just haven't got there yet.
I'm not familiar with the machine learning technique. Can you give just a brief description of it?
Brief would be difficult.
Yeah.
Essentially what it does is you start off with your entire SNP data set, it does a first pass and it kind of ranks all of the SNPs in terms of importance in that data set. And then you will cut out, say, the top 100 most important, and then it will go through it step by step, cutting out what it views as the least important each time, and looking at the amount of variation that the remaining SNPs are able to explain in your data set. Or what your data set in relation to whatever phenotype you've given it. Which I'm not sure, hopefully that's okay. We can discuss it, it's very complicated, it took me weeks to get it. Yeah, essentially you just take out the least important and then see if you can explain more of your phenotype based on the SNPs that we have left.